Hello everyone, welcome back to Infinity Learn by Sri Chaitanya NEET channel. My name is Kiran Chaudhary and I am your botany faculty at Infinity Learn. My dear students, in this class we will be discussing about all the important previously asked questions by NTA from your chapter number 10, class 11, that is cell cycle and cell division. Let me tell you, with respect to your NEET exam, your coming NEET 2022, this chapter holds prime importance. Every year, at least 2 to 3 questions are asked from this chapter. Chapter is quite conceptual in nature. So let's check out, let's find what are the questions that NTA has already asked. Let us also see and analyze what are the important paragraphs, concepts, lines and facts written in NCERT with respect to this chapter on which NTA is repeatedly asking question. Because not to forget guys, what is the characteristic of NTA? That it repeats history. NTA tends to repeat question or make those questions or rather I should say make questions on those facts or information on which it has already asked question, right? So it becomes quite important for each one of you who are aiming to clear or crack NEET 2022 to practice all the PYQs, important PYQs that NTA has asked in the history of NEET exam from 2013 to 2021. So this is what we are going to do in this class, my lovely kids, and I will really appreciate if you will participate in the session as well. So let's get started, kids. Before starting the session properly, I would love to say till now, if you haven't subscribed to Infinity Learn by Sri Chaitanya NEET channel, do consider subscribing it. Give me a thumbs up and also share this important session with all your friends so everybody get benefited. So let's start with the very first question. Yes, come on everyone in the class, try to answer this question, and trying is maximum that I am expecting from everybody in the class. So do not think whether your answer is going to be right or wrong. Doesn't matter. What matters is that you try it. If you're going to get the answer or question right, excellent, perfect. But if you get it wrong, I promise you, never again, at least till your NEET 2022 paper, you won't make this mistake. You will not make this mistake again. But for that, a little you have to do is try. So let me read out the question for you guys. Identify the correct statement with regard to G1 phase, also known as gap 1 of interphase. Remember kids, in a cell cycle, there are two important phases. Interphase, also known as preparatory phase, where cell is actually preparing itself for the division, and the actual division or the most dramatic phase of the cell cycle is the M phase, where karyokinesis and cytokinesis take place. So this interphase of the cell cycle, this preparatory phase, is also having subdivisions. It has G1, S and G2. So different activities takes place when a cell is in G1 phase, S and G2. So this question is about G1 phase. So out of the 4, you need to identify which of the following statement correctly states the activities a cell will undergo when it is under G1 of interphase. Any guesses? Okay, then let me read out for you. DNA synthesis or replication takes place. So this actually takes place, DNA synthesis or replication, in S phase. In S phase, my dear students, of interphase. So it's not the activity of G1. This is the activity of S phase. Now let us talk about reorganization of all cell components takes place. No. Cell is metabolically active but does not replicate its DNA. That's absolutely correct for G1. So cell is metabolically active. It is doing anabolism and catabolism, my dear students, to produce something or the other. But the point is, DNA replication, right, or copying of the genetic material is not taking place in the G1. It will take just after the G1 in S phase. So cell is metabolically active. Nuclear division takes place. Nuclear division means karyokinesis. Nuclear division, my lovely kids, means karyokinesis, and such kind of karyokinesis does not take place in interphase, it is the characteristic of M phase. It's not the characteristic of interphase. So that is why this is also wrong, and C is your correct answer. Cell is metabolically active, grows but do not replicate its DNA. Now let's see the next question, guys. The next question is some dividing cells. Okay, this is a quite good question. This question was asked in, if I remember it correctly, in NEET 2020. So some dividing cells exit the cell cycle and enter, NEET 2020, okay. So enter the vegetative inactive stage. This is the quiescent stage G0. This process occurs at the end of, I repeat myself, some dividing cells exit the cell cycle and enter vegetative and inactive stage. This stage is called quiescent stage, and this process occurs at the end of. So in the entire cell cycle, when exactly a cell decides not to continue division and enters into an inactive stage known as G0 stage or quiescent stage. So what do you think, when exactly a cell enters into G0? At the end of which phase, I repeat myself, at the end of which of the phase in a cell cycle, cell will try to enter into the G0? It's a very good question asked by NTA, and most of the students were unable to get it correctly. So in this session, I'm going to provide you right answer. Do understand why that particular answer is the correct one. But I'm not getting any reply as of now. Okay, fine. Feel comfortable in the class and do not think whether ma'am is going to judge your answer or not. I'm not here to judge your answer. I'm here to help you, my kids, to prepare for NEET 2022. Alright, so do not think like that. Just try to give answer. So the right answer is at the end of G1 phase. Right? So at the end of the G1 phase, the cell will exit. The cell will exit the G1 phase in order to enter the G0. But remember, in order to enter the G0, cell never exit or terminate the cell cycle. It just exit or terminate the G1 phase. This is very important, and even on this fact, NTA asked question that is going to come next, I guess. Okay, so I hope, please remember, this is a very important question. Next. Yeah, so as I told you, cell in G0 phase. When exactly cell enters into G0 phase or quiescent stage, what exactly it has done? So has it terminated the cell cycle, exited the cell cycle, entered cell cycle, or suspended the cell cycle? Any guesses? That's a beautiful question. So as I told you, in order to enter the G0 phase, I repeat myself, now listen this again carefully. In order to enter the G0 stage, whenever a particular cell decides that it do not or does not want to, hello Rajesh, very warm welcome, that's absolutely correct, excellent, suspend the cell cycle, right? So what I'm trying to say is, whenever a particular cell decides not to continue to divide further, right? So what it will do, it will enter into the G0 stage. When it will enter into the G0 stage? At the end of G1. And in order to enter the G0 stage or quiescent stage, a cell will exit the G1. That means at the end of G1, cell will exit the G1 to enter the G0, but in that whole process, cell is actually suspending the cell cycle. It is not terminating or exiting it. It is just suspending the cell cycle but exiting the G1, right? And please understand why the cell needs to suspend the cell cycle, so that in future, whenever this particular cell, which is right now in G0, needs to divide again in future according to the requirement, it can again enter into the cell cycle and undergo the division. But if it will exit the cell cycle, it won't be able to do that. So suspension means at a halt these cells are, but if it is a requirement, they will again enter the cell cycle and undergo division. I hope this is making proper sense. Do let me know if there is any single doubt. I would love to explain it in some other way if this is not helping you. Next question. Okay, Rajesh, can you try this question? Rajesh, I think nothing can be more simpler than this. Correct sequence of phases of cell cycle. Come on, give it a try, Rajesh. I don't think so anything can be more simpler than this. I don't think so anything can be more simpler than this question from cell cycle and cell division. Right? It is just asking about the sequence. Yes, Rajesh. Will you try it? Yeah, excellent, Rajesh. Quite impressive. So G1, then S, then G2 and then M. So this is your interphase and this is your M phase. It will also have karyokinesis and cytokinesis. Right? Excellent, Rajesh. Let's see the next question, Rajesh. So this is the question, Rajesh, and this was the diagram given. This was the diagram given in the NEET paper, right? And this is the hint. This wasn't given in your NEET exam, right? So this is the hint. You have to get the answer by using this diagram, but the hint is this. So try to relate this diagram with this diagram and try to get the answer. So this was the part of the actual question and this is the hint I am providing. So this was how you were supposed to identify the right answer. Come on, Rajesh, tell me, what do you think is the right answer? Given below is a schematic breakup of the phases or stages of cell cycle. Which of the following is the correct indication of the stage or phase of the cell cycle? So what does this A, B, C and D is indicating is actually the question. So I'll be side to this so that you can easily correlate. Yes, Rajesh, will you try this? Okay, then let me help you out over here. So see this part is your, let me change the ink then, this is your G1, this D is your S phase, this one is your G2, this whole is, my dear students, your M phase, and in N phase this A is your karyokinesis. Now the C represents to cytokinesis, this represents to prophase, metaphase, B represents to anaphase and this represents to telophase, right? So if you notice, what is the right answer? So C karyokinesis, no, C is cytokinesis. C is not karyokinesis, C is your, I hope you are able to see this, C is your cytokinesis. D is the synthetic phase, yes, D is your S phase is also known as synthetic phase, that's absolutely correct. A is cytokinesis, A is not cytokinesis, A is actually your karyokinesis, A is karyokinesis. Next, B is metaphase, no, B is not metaphase, B is anaphase. Is this okay, Rajesh? Are you happy with the explanation? Any doubts? Do let me know in the chat box, Rajesh. Okay, then let's take the next question. This was the question that was asked in the latest NEET exam, NEET 2021. So you were supposed to match this and you can take the example of this. This is hint. This is also hint. This wasn't the part of the actual question. So tell me, what do you think is the right matching? Try this, Rajesh. Come on, guys. Try this one. Try this one, Rajesh. Are you trying or shall I help you? Okay then. So let me help you then. Let's talk about S phase. So what happens in S phase? DNA replication. What happens in S phase is your DNA replication. In what is a quiescent stage? Quiescent stage is an inactive stage. Right. What happens in G1 phase? G1 phase is the time interval. G1 is the time interval between an M phase and another cell, or between an M phase and DNA replication. So interval between mitosis means it's talking about the M phase, and initiation of DNA replication. So this is a characteristic of G1, and G2 is where the proteins are synthesized for the cell division to take place. Is this okay? Is this okay now, Rajesh? Any doubts in this question? If there is any single doubt, do let me know, Rajesh. Okay then. Fine. So I'm going to take another question. Okay, so this was also asked in NEET 2021. So I'm leaving you with this question, and just give me a moment. I need to take a sip of water and try this question. It was asked in NEET 2021. Okay, just try this one. Just coming. Okay guys, try this question. So let me read out the question for you. The fruit fly has 8 chromosomes. Here 8 chromosome is equals to 2n. That means this fruit fly in a diploid cell, of a fruit fly, the number of chromosome 2n equals to 8. During interphase of the mitosis, if the number of chromosomes at G1, right my kids? So this is quite simple, and let me help you out how to get the answer. So listen this carefully. In S phase, there is a property of S phase that in S phase what happens? Amount of DNA doubles. Amount of DNA doubles. Now let's talk about number of chromosome. Number of chromosomes. So if I talk about number of chromosomes, they remain same. Right? So the number of chromosomes that were there in the fruit fly cell, in a diploid fruit fly cell in G1 phase will be the same. Do let me know if there is any doubt, any single doubt in the chat box, my lovely kids. So let's see the next question then. Which of the following options gives the correct sequence of events during mitosis? Which of the following options gives the correct sequence? Which of the following options gives the correct sequence of events during mitosis? Come on Rajesh, give it a try. Come on Rajesh, give it a try, my lovely kid. Koshish to karo. Okay, then let me help you out. First of all, what will happen? First of all, in mitosis, in the M phase of the mitosis, which phase comes? Prophase comes. So these two are condensation and nuclear membrane disassembly. This will take place in the prophase. So remember what happens in mitosis M phase. First there is prophase, then there is metaphase, anaphase, telophase, right? So what is going to happen in the prophase? Condensation of chromatin material into chromatids to form chromosome will take place. At the late prophase, the nuclear membrane will disintegrate, right? Next, in metaphase, what will happen? Arrangement of the chromosomes at the equator will take place, right? So this is a characteristic of metaphase. Now the next phase is anaphase. So in anaphase, what will happen? Centromere will split. As a result, the sister chromatids will separate out to form daughter chromosome, right? So splitting of the centromere and segregation of the chromatids as daughter chromosome towards the two opposite pole will take place. So these two are characteristics of anaphase. And last one is the telophase, right, where the activities that were happening in prophase will get reversed. Is this clear? Any doubts till now? So the answer is A. Do let me know if you're facing any single difficulty in answering or understanding any single question, as well as the explanation and solution. Okay, so let's take another question. Spindle fibers attach on where exactly during M phase the mitotic spindle, right? You remember the mitotic spindle that you see. So where exactly over a chromosome these spindle fibers are attached? What is the attachment point of these spindle fibers on a chromosome? Any guesses, Rajesh and my other kids? Come on, give it a try. Give it a try. Okay. Spindle fibers attach onto the kinetochores of the chromosome. They attach to the hello uncle, very warm welcome to the class. Excellent, excellent. So they attach to the kinetochores of the chromosome. Kinetochores are the structures that are present over the centromere. Excellent. Let's see another question then. If you are provided with a root tip of onion in your class and you are asked to count the chromosome, that means whenever your aim or objective is to count the number of chromosome or to see the number of chromosome in a cell, in a dividing cell, then which of the following stage will be the most convenient one? Good afternoon, uncle. So tell me, uncle and Rajesh, what is the right answer to this? Out of these four stages, which one is more convenient to check out the number of chromosome? Uncle and Rajesh, there are two things, number of chromosomes and shape of chromosome. So whenever it is the case of number of chromosome, go with metaphase. Whenever they are asking about shape of chromosome, whether it is V shape, L shape, J shape, I shape, yeah, so it is what? It is the anaphase. So whenever you want to see the shape of chromosome, anaphase. Number of chromosome, metaphase. Is this clear, uncle and Rajesh? Is this clear, uncle and Rajesh? Clear hai? Ab clear hai bachche? Yeah. Now let's see the next question. Centrioles duplicate in. Come on, give it a try. Centrioles duplicate in. Give it a try, my lovely kids. Centrioles duplicate in. Come on, centrioles duplicate in. Give it a try. Centrioles duplicate in. Yeah, that's absolutely correct. Centriole duplicates in S phase. So during S phase, DNA duplicates or replicates in the nucleus, whereas centrioles duplicate or replicate in cytoplasm, right? So will you remember this now, Rajesh? Will you remember this, Rajesh? Let's see another question. In S phase of the cell cycle, what will happen? This is the most simple question. Nothing simpler than this can be asked from S phase, right? Pretty simple question. Give it a try, my kids. Give it a try. In S phase of the cell cycle, which of the following activities taking place? Or rather I can say, better I can say, what is the characteristic of S phase? Yes, uncle? Yeah, what about Rajesh? Will you give it a try, Rajesh? कोशिश कीजिए राजेश. कोशिश और प्रयत्न ही है जो मैं आपसे इस सेशन में उम्मीद कर रही हूँ. Yes, Rajesh, good. Amount of DNA in each cell doubles, but number of chromosomes remain same. बट याद रखना क्रोमोसोम्स पर 2 क्रोमैटिड्स आ जाएंगी, सिस्टर क्रोमैटिड्स आ जाएंगी, बिकॉज सेल में डुप्लीकेशन हुआ है डीएनए का. सो बट नंबर ऑफ क्रोमोसोम रिमेन सेम. नेक्स्ट, that's a beautiful question, that's a very good question. Please try this question, Ankul and Rajesh. A somatic cell. What is a somatic cell? Somatic cell means a diploid cell. Somatic cell is a diploid cell. A somatic cell that has just completed the S phase, यानी S phase complete हो गया है, that means amount of DNA double हो जाएगा, number of chromosomes same रहेंगे. So a somatic cell that has just completed the S phase of its cell cycle as compared to the gametes of the same species. Gamete कैसे होते हैं? Haploid होते हैं. Gametes कैसे होते हैं? Haploid होते हैं. Right? और diploid cell में number of chromosomes कितने होते हैं? Number of chromosomes equals to 2n. And amount of DNA in a diploid cell. A somatic cell is a diploid cell. I repeat myself. Somatic cell is a diploid cell. So in a somatic cell, number of chromosome 2n and amount of DNA. Amount of DNA कितना रहेगा? 2c, right? And what is a gamete? Haploid. यानी number of chromosome n and amount of DNA c रहेगा, right? So it is just half. Number of chromosome and amount of DNA is just half in a gamete in comparison to a somatic cell. Now you were supposed to find out amount of DNA and number of chromosome in this somatic cell when it has completed the S phase. So what activities will take place during S phase? Amount of DNA double हो जाएगा but number of chromosome remains same. So let's see what will happen to this diploid cell, right? यहाँ देखते हैं हम लोग. ये था हमारा diploid cell before S phase. Before S phase, in a somatic cell, in a diploid somatic cell, number of chromosome कितने होंगे? Number of chromosome equals to 2n. Amount of DNA कितना होगा बच्चों? Amount of DNA, amount of DNA 2c. Right? Now, this is the first situation. Now, let's see the next situation. After S phase. After S phase क्या होगा? In a diploid somatic cell. Right? So, what happens in S phase? Amount of DNA double. Number of chromosome remains same. Excellent. So excellent, very good, Ankul and Rajesh. Number of chromosomes will be same 2n, but amount of DNA will be 4c. Okay, amount of DNA is 4c. Now compare this data. My kids, compare this data. कौन सा कलर दिखेगा इस पे? ब्लू देखना चाहिए. Now compare this data with a haploid cell. तो एक haploid gamete cell में, in a haploid gamete cell, what is the number of chromosome n and amount of DNA कितना होगा? Amount of DNA is going to be number of chromosome n and amount of DNA will be C. Now compare this data. Now compare this situation of this diploid somatic cell which has completed S phase. Now compare this situation with the haploid gamete cell. So what you can see, the number of chromosomes that are present in this diploid somatic cell after S phase will be double that of the gamete, but amount of DNA is going to be 4 times, because here it is C, here it is 4C, here it is N, here it is 2N. So amount of DNA 4 times, number of chromosomes double in comparison to a haploid gamete. So is this clear, my dear students? I really hope after this whole explanation this question becomes very simple for you. Is this clear now? Uncle, will you make this mistake again? Rajesh was correct. Thank you, I appreciate that. But uncle, say, will you make this mistake again? So the answer is A, twice the number of chromosome and 4 times the amount of DNA. Will you make this mistake again? The point, the whole objective of taking this class is not to repeat the mistakes again, right? क्वेश्चन पूरा नहीं पढ़ा अंकल, क्वेश्चन पूरा नहीं पढ़ोगे तो कैसे चलेगा? कहाँ जाना है भाग के, बताओ अंकल? अभी अपनी कोई ट्रेन नहीं छूट रही है और एग्ज़ाम में भी कोई ट्रेन नहीं छूटेगी. अगर हम इसी तरीके से गलत पढ़ते जाएंगे तो एग्ज़ाम और उसके आप अपना जो टारगेट है उससे दूर होते जाएंगे. तो हमें इस तरीके की कोई जल्दबाजी नहीं दिखानी है. ऐसी जल्दबाजी दिखाकर हम कहीं नहीं पहुंच पाएंगे. राइट अंकल? प्रॉमिस मी इन दिस सेशन every question that you're going to attempt either in mock test or in your NEET 2022, you will read it carefully and calmly. नंबर ऑफ क्रोमोसोम में फेज में दिखता है मैम, नंबर ऑफ क्रोमोसोम एम फेज में दिखता है. येस, दैट्स एब्सोल्युटली करेक्ट. तो ये नंबर ऑफ क्रोमोसोम अब अभी आपको दिखेंगे नहीं. क्यों? क्योंकि अभी तो डीएनए क्रोमैटिन मटेरियल या क्रोमैटिन फॉर्म में प्रेजेंट है अंकुल. और ये जो प्रोफेज आएगा एम फेज का, राइट, तभी तो उसका कंडेंसेशन स्टार्ट होगा, क्रोमैटिड बनेगा, क्रोमोसोम बनेगा. तो ये दिखेगा नहीं. ये डबल हुआ अमाउंट, ये जो अमाउंट ऑफ द डीएनए दैट इज डबल्ड इन द एस फेज, दिस इफेक्ट विल बी सीन in M phase. In the late prophase or in early metaphase, you can see the compact chromosome with two sister chromatids. तो ये two sister chromatid कहाँ से आई? Because DNA replicate कर गया था S phase में. Is this clear, Ankul and Rajesh? Is this clear, Ankul and Rajesh? So Ankul, अब से आराम से क्वेश्चन पढ़ना है, नो जल्दबाजी. Okay, next question. Anaphase promoting complex. अब ये देखो, this question was not directly given in NCERT, but ये NEET का क्वेश्चन है. इसे NEET एग्जाम में पूछा गया था. if I'm not wrong, ये 2016 का क्वेश्चन है, right? So anaphase promoting complex is a protein degradation machinery necessary for proper mitosis of animal cell. If APC is defective in a human cell, which of the following is expected to occur? Now listen this, definitely the information was not given in your NCERT, but फिर भी आप इस क्वेश्चन को करेक्टली अटेम्प्ट कर सकते हैं. but still you can attempt this question without any error if you use your common sense. Now how to use your common sense in such question whose information you are reading for the first time? Try to read the question. NTA gave you the hint. NTA ने क्वेश्चन में ही हिंट दिया है कि APC क्या है, एक इम्पॉर्टेंट या एक इम्पॉर्टेंट मशीनरी है जो प्रॉपर तरीके से माइटोसिस होने में हेल्प करती है, राइट? यानी कि एनाफेज प्रमोटिंग कॉम्प्लेक्स, वो एनाफेज को प्रमोट कर रहा है. और एनाफेज में क्या होता है? सेंट्रोमियर में स्प्लिटिंग होती है, सिस्टर क्रोमैटिड सेपरेट हो जाते हैं और डॉटर क्रोमोसोम में कन्वर्ट हो जाते हैं. सो लेट्स से इन अ मेटाफेज हमारे पास क्रोमोसोम कैसे था? इस तरीके से था, right? This is how the chromosome is in metaphase, all the chromosome. Now what happens when there's a transition from metaphase to anaphase? What happens when a transition is from metaphase to anaphase? Now this is irritating me. तो मेटाफेज से जब हम एनाफेज की तरफ जाएंगे तो क्या होगा अंकुल एंड राजेश एंड अदर किड्स? सेंट्रोमियर में स्प्लिटिंग होगी, सेंट्रोमियर में स्प्लिटिंग होगी. तो सेंट्रोमियर के स्प्लिट होने से ये जो दो सिस्टर क्रोमैटिड्स हैं, these two sister chromatid will separate out and as a result there will be formation of two daughter chromosomes जो कि opposite poles पे चले जाएंगे. This is what happens in anaphase. So if this complex name is anaphase promoting complex, तो ये क्या करेगा, ये anaphase को promote करेगा, centromere की splitting को promote करेगा ताकि segregation हो पाए, डॉटर क्रोमोसोम्स बन पाए. बस यहीं से तो आंसर अब पता लगा करना था. सो अगर आप बस अपना कॉमन सेंस यूज करेंगे, पैनिक नहीं करेंगे, यू विल बी एबल टू अटेम्प्ट सच क्वेश्चंस आल्सो. सो व्हाट डू यू थिंक इज द राइट आंसर? डेफिनेटली क्रोमोसोम्स विल नॉट सेग्रीगेट. व्हाई? क्यों? क्योंकि एनाफेज प्रोमोटिंग कॉम्प्लेक्स अगर डिफेक्टिव हो गया है, व्हाट इज द क्वेश्चन? क्वेश्चन इज एनाफेज प्रोमोटिंग कॉम्प्लेक्स इज नेसेसरी फॉर efficient mitosis or correct mitosis to take place. Now if in an animal cell this APC is not working properly, then what will happen? The chromosome will not be able to segregate because this anaphase won't be promoted, the segregation won't be promoted. Pretty simple, yeah? Okay, so this is how you can solve the question with common sense. Now I would also like to provide you little more information about anaphase promoting complex, right? So you can note this. What is anaphase promoting complex? This anaphase promoting complex actually produces two protein components. Let's talk about first protein component, that is protein component 1. So I am giving you now detail of this complex, but to attempt that question you are not supposed to. Video is stuck. Okay, just give me a moment. I'm just coming back. Just give me a moment. वीडियो अटक गया है. Is the video working now? Uncle and Rajesh. Now okay, perfect. So as I was talking, my dear students, you did not know that this much information to answer the question you can give by simple common sense, but let's try to understand over here what this APC is all about in detail. APC produces two types of protein component. This is protein component 1, this is protein component 2. Protein component 1 do the inactivation of securin, and as a result separation of chromatids, that means centromere splitting takes place and the sister chromatids separate out as daughter chromosomes. Now if you know, if this APC is not working, what will happen? Inactivation of securin will not happen, and as a result separation of chromatids will not take place. So this was the information over which NTA asked the question. Now let's talk about another protein component 2 that is produced by APC. It leads to an inactivation of mitotic cyclin, which helps in reassembly of the nuclear membrane. That will happen later after anaphase, right? Is that okay? Yeah. Now let's see the next question. How many mitotic divisions are needed for a single cell to make 128 cells? Come on. How many mitotic divisions? It's pure simple maths. Pure simple maths. Come on guys, give it a try. Uncle and Rajesh, would you like to give it a try once? You can give it by 2 raised to the power n equals to 128. Let me help you out then. So how to answer these questions? So in mitosis, what happens? In mitosis, my kids, from one cell, at the end of mitosis, you get 2 cells, right? So from after one mitotic division, you get from one parental cell 2 daughter cells, right? So as a result, in order to in order to find out total number of divisions that are required to produce 128 cells, you will use the formula 2 raised to the power n equals to 128, right? So this is how, but this formula to be applied in mitosis only, right? Because it is leading to the production of 2 cells. Now, important, what is important over here is, my dear students, now you need to factorize 128. Now see, by multiplying how many times 2? How many times 2 are multiplied in order to get 128? To 2 raised to the power 7 is 128, is 128. Hence, when 7th time mitosis will take place, there will be production of 128 cells. नो 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 नो, तो जितनी बार बनते जाएंगे वो सारे भी तो बार बार डिवाइड करते जाएंगे अंकुल। एक बार में कितने बनेंगे? 2, अगली बार में कितने बनेंगे? वो 2 फिर से डिवाइड करेंगे, राइट? वो 4 फिर से डिवाइड करेंगे, इस तरीके से ये चलता ही जाएगा। नेक्स्ट क्वेश्चन, mitotic anaphase differs from metaphase in possessing. Mitotic anaphase differ from metaphase in possessing. Come on, give it a try this, uncle. Hello Saira, very warm welcome in this session. Give it a try, my lovely kids. That's fine, Saira. Okay, so mitotic anaphase differs from metaphase in possessing double the number of chromosomes and same number of chromatids. How? So let's take an example of first metaphase. So how chromosomes looks like in metaphase? How chromosomes looks like in metaphase? This is metaphase, and how they will look like in anaphase? In anaphase, the splitting will happen, right? The splitting of centromere will happen, and as a result, there will be production of daughter chromosomes. Right? So see here, how many chromosomes are there? Number of chromosomes? 3. Number of chromosomes? Number of chromosomes are 3. Number of chromatids? Number of chromatids. Chromatids का number कितना होगा? 1. Each chromosome in a metaphase is having 2 chromatids, 2 sister chromatids. So total number of chromosome are 3. So number of chromatids will be 6. Now see over here, after the splitting of the centromere takes place, number of chromosomes कितने हैं? 1, 2, 3, 1, 2, 3, 6 chromosomes. But number of chromatids will be 6 only again. So if you notice, from anaphase and metaphase, what is the difference? Number of chromosomes increases, but number of chromatids remain same. Why? Because these number of chromatids, why they will be same, my dear students? Because the new 3 chromosomes that has been produced were produced by splitting of the centromere. So the sister chromatids that were there in the metaphase, now they will be acting as a daughter chromosome. Is this okay, uncle? Saira, Rajesh, is this okay, my lovely kids? Let's take the another question then. Dissolution of okay, these were the important question that that were asked by NTA NEET exam from mitosis topic and cell cycle. Now let us talk about important questions from meiosis, right? So this is the very first question from meiosis. Dissolution of synaptonymal complex occurs during pachytene, zygotine, diplotin, leptotin. What do you think is the right answer? Uncle, Saira, come on guys, give it a try. Uncle, Saira, give it a try. Yeah, that's absolutely correct. So this synaptonymal complex was formed in zygotine stage when two homologous chromosomes were coming together so that they can undergo crossing over in the pachytene stage. Now after pachytene, in pachytene the crossing over is completed, right? So now these two homologous chromosomes do not need to be remain to stay together. So now from diplotin onward they will start separating out. But remember, the separation of two homologous chromosomes starts in diplotin of prophase 1 but it ends in the anaphase 1. That's a very important concept on which NTA has also asked question. Next question. Identify the meiotic stage in which the homologous chromosomes separate while the sister chromatid remain associated at the centromere. So you need to find out of these four in which stage the homologous chromosome separates but splitting of the centromere or separation of the sister chromatid does not take place. Any guesses on this? Uncle, Saira? Any guesses on this? Uncle, Saira, Rajesh? Uncle, Saira, Rajesh? That's absolutely correct. In anaphase. Why? Because no splitting of centromere takes place. That's the reason why sister chromatid still remain attached to the centromere and only the homologous chromosome that came together during zygotine of prophase 1 will finally get separated out. Remember, the separation started in diplotin of prophase 1. Next question. Crossing over takes place, I repeat myself, crossing over takes place between which chromatids and in which stage of the cell cycle? So here are the options. Come on, give it a try, my kids. Crossing over takes place between which chromatids and in which stage of the cell cycle? Uncle, Saira, Rajesh, give it a try. Give it a try, my kids. That's absolutely correct. They will take place between non-sister chromatids of homologous chromosome at pachytene stage. So how does it look like? So let me show you. Let's say this is your one chromosome and let's say this is your another chromosome, right? So crossing over will take place here, right? So what are these? Let me show you with another color. These two are sister chromatids. These two are also sister chromatids. But when I talk about this and this, these are non-sister chromatids, right? These two are sister, these two are also sister, but these two are non-sister chromatid, and crossing over is taking place between non-sister chromatid, and in which stage? In pachytene stage. with the help of recombination nodule. That's right, uncle. Now let's talk about another question. Here comes another question. The stage during which separation of paired homologous chromosome begins. So see, I have already repeated this question multiple times. So let me see who can answer this correctly this time. The stage during which separation of paired homologous chromosome begins is. Come on guys, tell me what is the right answer. In which stage these homologous chromosomes actually started separating out? Everybody in the class, Uncle, Saira, Rajesh, give it a try. Give it a try, my lovely kids. Yeah, that's absolutely correct. The separation starts in diplotin stage. The separation begins in diplotin. So, separation begins in diplotin. But the separation of homologous chromosome finishes or ends in anaphase 1. So, if you are unable to get this, please write it somewhere in your NCERT. The separation of homologous chromosome begins in diplotin and it ends in anaphase 1. And when exactly this pairing of homologous chromosome took place, this synapses took place during zygotine stage of prophase 1, right? Good. Next question. Match the stages of meiosis in column 1 to their characteristic feature. Yeah, so match this. Try to give me the answer, my kids. Yeah, so what do you think is the right answer? Okay, so in packaging what will happen? Crossing over takes place. In metaphase 1 what will happen? Chromosome will align at equatorial plate. Diakinesis is known for terminalization of chiasmata and zygotine is known for pairing of the homologous chromosome. So A pairs with 3, B pairs with 4, C pairs with 2 and D pairs with 1. So A is the right answer. Tell me now, is this okay? Yes, I got it, uncle. Thank you. Next question. Next question is about, in meiosis crossing over is initiated. So where exactly crossing over starts? Where exactly crossing over starts? Very simple question. Tell me, uncle, where exactly the crossing over is starting? Come on. Where exactly the crossing over is starting? Yeah, so crossing over starts at packaging, and crossing over not even starts at but even finishes in packaging, and that is why in diplotin stage, the next to packaging, the segregation or separation of homologous chromosomes also starts. Next, start bhi khatam bhi. Yes uncle, you remember it rightly from the class. Okay, crossing over initiated at packaging. Next question, arrange the following events of meiosis in correct sequence. अब आपको ये events बताने हैं. That's a pretty good question. Let me see if you can arrange it correctly. These are the sequences and remember all these are from prophase 1. ये सारे prophase 1 के ही हैं. So can you arrange them? These all are the events of prophase 1 of meiosis 1. So can you arrange them correctly? Uncle? क्या आप इसे सही तरीके से अरेंज कर सकते हैं? ज़रा कोशिश तो कीजिए. Okay, then let me see it. So the first one should be synapses that takes place in zygotine. So what is synapses? When homologous chromosomes comes together. Okay, then is your crossing over that will take place in packaging. And then terminalization of chiasmata in diakinases, and then disappearance of the nucleolus, that will be in the late prophase, towards the end of the prophase, towards the end of these, towards the end of these, towards the end of the diakinases, this will take place. So this should be your 1, this should be your 2, 3 and 4. So answer is 2, 1, 3 and 4. Yeah, you are correct, uncle. Next question. 2, 1, 3 and 4. So enzyme, yeah, the enzyme that you were talking about, recombinase. The enzyme recombinase is required at which stage of the meiosis? Pretty simple for you now. Enzyme recombinase is required at which stage? Yes, uncle. Enzyme recombinase is required at which stage? That's right. Recombinase enzyme is required for recombination to take place in p-chitin. Next question. The complex formed by a pair of synapsed homologous chromosome is called bivalent equatorial plate kinetochore. What is the complex called which is formed by two homologous chromosomes or synapsed homologous chromosomes? Any guesses? Yes, uncle, give it a try. So this is your synapsed homologous chromosome, right? So what do you call it? You can either call it as a bivalent, or you can also call it as tetrad. So what is the characteristic of it? How many chromatids are there in a bivalent or a tetrad or a synapsed homologous chromosomes? A pair of homologous chromosomes which are synapsed in zygotine. They have actually 4 chromatids, 2 each. Chromatid 1, 2, 3 and 4. These 2 are sister, these 2 are sister, these 2 are non-sister, in which crossing over will take place in the p-chitin stage. So how many chromatids are there? 4 chromatids are there. And how many centromeres are there? How many centromeres are there? 2 centromeres are there. That is why according to the centromere it is bivalent, and according to chromatid is tetrad, right? 2 centromere and 4 chromatids. 2 centromere and 4 chromatids. Is this clear, uncle? Yeah, zygotine banega. Is this clear, uncle and my other kids? So the right answer is bivalent. Is this clear, uncle? Can we move to the next question? Next question is, synapses occurs between. See, it's very simple. So, okay, you can see the related questions NTA is repeating. NTA just framing or framing the same question in a different way. They're changing the language, that's it. So, synapses occurs between. Synapses occurs between. Very simple, pretty simple. Synapses occur between, uncle. Synapses occur in between. That's right, 2 homologous chromosomes in zygotine stage. When paternal and maternal chromosomes change their materials with each other in cell division, this event is called bivalent forming, dyad forming, synapses, or crossing over. What do you think is the right answer? So remember, out of the 2 homologous chromosomes, one you get from the male father, one you get from the father and one you get from the mother. So yeah, that's right. So exchange means crossing over. Excellent. Next question. Which statement best explains the evolutionary advantage of meiosis or significance of meiosis we are talking about? So what do you think is the best one that explains the significance of meiosis with respect to evolution or has an evolutionary aspect in it with respect to meiosis? Come on guys, give it a try, uncle. Give it a try to this question, uncle. Yeah, that's right. Genetic recombinations are possible from generation to generation. Yeah, that's right. So, genetic recombinations are possible from generation to generation that leads to variation, and variations are the base of evolution to take place. Excellent. Because in meiosis 1, in prophase 1, in packaging, crossing over is taking place between non-sister chromatids of homologous chromosome. As a result, this leads to recombination of the genetic material. That's right. Next question, uncle. Meiosis 2 performs. In meiosis 1, so meiosis has actually two parts, meiosis 1 and meiosis 2. So how do you see meiosis 2? How do you see meiosis 2? Come on. Which of the following option matches with the activity that takes place in meiosis 2? Give it a try. That's right, separation of chromatid. Meiosis 2 is equivalent to mitosis. So whether it is meiosis 2, whether it is anaphase 2 or anaphase of mitosis, separation of chromatids will take place in anaphase 2 because of splitting of centromere. So splitting of centromere takes place in anaphase 2 of meiosis 1, and hence the hence the sister chromatids separate out to form daughter chromosomes. Next question. Number of chromatids in, okay, I guess this is the last question of this session. Number of chromatids in each chromosomes at metaphase is, number of chromatids in each chromosomes at metaphase is. These are the options. Come on, uncle, that's the last question. Give it a try, uncle. Give it a try, uncle. Yes. Actually, uncle, the answer is whether it is mitosis or meiosis, whether it is metaphase of mitosis or metaphase 1 or 2 of meiosis, the number of chromatids in chromosome will always going to be 2, right? Because let's talk about mitosis. The chromosome will have 2 chromatids. In anaphase, it will split the centromere. Let's talk about meiosis. In metaphase 1, the chromosomes will have 2 sister chromatids. Even in anaphase 1 also, it will not split. Let's talk about meiosis 2. In meiosis 2, in meiosis 2 also there is metaphase 2 and anaphase 2. Even again, in metaphase 2 of meiosis 2, the chromosome will have, each chromosome will have 2 sister chromatids, right? But actually in anaphase 2 of meiosis 2, there will be splitting of the centromere and the 2 sister chromatids separates to form daughter chromosome. Is the explanation okay for you, uncle? Did you understand why this is the answer? Please let me know in the chat box, uncle. Okay, fine. So yeah, that's all, my dear students. So till now, if you haven't joined Infinity Learn Telegram channel, do join it to get assignment notes, daily updates, and the link is in the description. Thank you very much guys for listening to me. Thank you very much for participating in this session. मेटा 1 में तो डबल होते हैं, मेटा 1 में डबल होते हैं, मेटा 2 में भी डबल होते हैं, क्योंकि Na2 में जाकर सेंट्रोमियर स्प्लिट होगा। तो चाहे मेटाफेज कोई भी हो, क्रोमोसोम हर टाइप की मेटाफेज में, चाहे वेदर इट इज माइटोसिस और मियोसिस, इन ऑल टाइप्स ऑफ मेटाफेज each chromosome will have 2 chromatids. Is this okay, uncle? So thank you guys for listening to me. If you haven't subscribed to Infinity Learn NEET channel, do consider subscribing, like, share and press the bell icon so that whenever I'll go live, you won't miss any single session in future. Thank you very, very much for joining this class. That's all from this class.